Hello, myself Rajendra Shetty, Assistant Professor at Ganpat Parsikar College of Education, Harmal, Goa. Title of the unit, Biomolecules. Module name, Structure of bDNA, alternate forms of DNA, that is A, C, D and Z. Outline, structure of bDNA, other forms of DNA, that is A, C, D and Z forms of DNA. Learning outcomes, elucidates the structure of bDNA, differentiates other forms of DNA, that is A, C, D and Z. Structure of bDNA is also referred as balanced DNA. This model was proposed by Watson and Crick in 1953. As per this model, DNA consists of two helical chains coiled around the same axis in the right-hand direction. Two chains in helix run anti-parallel to each other. Diameter of helix is 20 angstrom. Helix makes one complete turn at every 34 angstrom. Internucleotide distance is 3.4 angstrom. Each turn consists of approximately 10 nucleotides. Helix has two external grooves, that is major groove and minor groove. As you can see in Watson and Crick's model of DNA, you can clearly see the two helical coils of the DNA. They are wound around each other in right-hand direction and they run anti-parallel to each other in 3' to 5' direction and 5' to 3' direction. One complete turn, it takes 34 angstrom and the distance between the nucleotide is 3.4 angstrom. Also, it shows presence of major groove and minor groove. Composition of DNA. DNA is a biopolymer of deoxyribonucleotides. It is made up of three important molecules, that is phosphate group, deoxyribose sugar and nitrogenous bases. Nitrogenous bases present are of two types, that is purines and pyrimidines. Purines are of two types, that is adenine and guanine. Pyrimidines present are cytosine and thymine. As far as comparison with RNA is concerned, it differs from RNA in presence of the type of pentose sugar, that is, here there is presence of deoxyribose sugar and in case of RNA it is ribose sugar. As far as nitrogenous bases are concerned, nitrogenous bases adenine, guanine and cytosine remain same for both RNA as well as DNA, but in DNA thymine is present, whereas in case of RNA it is replaced by uracil. Backbone of DNA. Each DNA strand is a biopolymer of deoxyribonucleotides. Adjacent deoxyribonucleotides are linked by phosphodiester bonds. 5' carbon of deoxyribose of one nucleotide is linked with 3' carbon of deoxyribose of other nucleotide by bonding with phosphate group. Thus, hydrophilic backbone of alternating deoxyribose and negatively charged phosphate group remains on outside of helix. Here, you can clearly see the structure wherein a phosphodiester bond is shown between the two deoxyribonucleotides. Nitrogenous bases of DNA. DNA shows presence of two types of nitrogenous bases, that is purines and pyrimidines. Altogether, purines are of two types, that is adenine and guanine, and pyrimidines are of two types, that is cytosine and thymine. Carbon 1 of deoxyribose sugar is joined to the 9th position of nitrogen of purine, which you can clearly see in deoxyguanosine example, wherein the deoxyribose sugar, carbon number 1 and nitrogen of guanine is joined to each other. Carbon 1 of deoxyribose sugar is joined to first position of nitrogen of pyrimidine, which you can clearly see in the structure of deoxycytidine. Hydrophobic purines and pyrimidines remain on inner side of double helix. Hydrogen bonding between nitrogenous bases. Double helix is stabilized by interchain hydrogen bonding between purine and pyrimidine. Purine base pairs by hydrogen bond only with the pyrimidine base. Always, adenine pairs with thymine by 2 hydrogen bonds and guanine pairs with cytosine with 3 hydrogen bonds. The base pairs, adenine and thymine, and guanine and cytosine are known as complementary base pairs. Due to this, chains are complementary to each other. You can clearly see the hydrogen bonding between nitrogenous bases of the complementary strand in this given structure. The thymine bonds to adenine by 2 hydrogen bonds and guanine bonds with cytosine with 3 hydrogen bonds. Alternative forms of DNA. For about 20 years after discovery of DNA double helix in 1953, DNA was thought to have only B form with exactly 36-degree helical twist between its adjacent base pairs and a uniform helix geometry. Later, experiments found that DNA is polymorphic in nature and can show different forms depending upon physiological condition of cell. Even different parts of DNA can be in different forms. The different forms of DNA are aDNA, cDNA, ddNA, and zDNA. aDNA. When physiological condition shows 75% humidity and presence of sodium, potassium, and calcium ions, we get aDNA form. It is shorter and wider and shows 11 base pairs per turn. Rotation per base pair is plus 32.7 degree and is right-handed. Vertical rise per base pair is 2.56 angstrom and helical diameter is 23 angstrom. Due to this displacement, the depth of major groove increases and that of minor groove decreases. The A form is a metastable and quickly turns to the D form. C. DNA. When physiological condition shows 66% humidity and presence of lithium ions, we get C form of DNA. It shows 9.33 base pairs per turn. Rotation per base pair is plus 38.6 degree and is right-handed. Vertical rise per base pair is 3.32 angstrom and helical diameter is 19 angstrom. The base pair shows pronounced negative tilt by 7.8 degrees. D. DNA. The D form of DNA is found very rarely. Total number of base pairs per turn of helix is 8. Therefore, it shows 8-fold symmetry. This form is called as poly-DADT and poly-DGDC form. There is pronounced negative tilt of base pairs by 16.7 degree as compared to C form. That is, the base pairs are displaced backwardly with respect to the axis of DNA helix. zDNA. It shows zigzag structure. When physiological conditions show very light salt condition, we get Z form of DNA. It shows 12 base pairs per turn. Rotation per base pair is minus 30 degree and is left-handed. Vertical rise per base pair is 3.71 angstrom and helical diameter is 18 angstrom. Adjacent sugar residues have opposite orientation. One complete turn of helix is 45 angstrom long. Bases are more closer to axis. These are the comparative structural models of A, B, and Z DNA. These are the references. Thank you.